What's up, y'all? Professor Hurley here, bringing you another episode of Purple Bear Biology. In this episode, we are going to discuss some of the fascinating ways that species separate into different groups and eventually develop into separate species altogether. This process is referred to as macroevolution and often occurs when a population becomes fragmented and then isolated from one another. While in our previous conversation we talked about microevolution, which focuses on allele frequency changes over time, for macroevolution we're going to be looking at how large populations change into different groups over time. Before we get started on how a species evolves, let's first figure out what a species is. How in the world do scientists determine what is one species or two distinct species? It can be much more challenging than you think. For example, look at the songbirds here in the left. They're pretty similar, right? Indeed, they do have many similar morphological characteristics, but if you were to put them in the same habitat, they have specific characteristics, primarily behaviors, that inhibit them from reproducing. Of course, then if we look at this crazy species down here in the right, we can see that it's incredibly diverse with lots of different characteristics. And yet despite looking so different, all of these are the same species. So how do we figure this out? For the purpose of this conversation, we are going to be using something called the biological species concept. Essentially, in order for organisms to be considered the same species, they must be able to interbreed and produce fertile offspring. Disruption in a group of organisms' ability to interbreed is normally a result of two populations becoming isolated and having no gene flow between them. I should mention that the biological species concept is not perfect. For example, we cannot use it to compare fossils because dead things don't have sex. We also cannot use it for organisms that reproduce asexually like bacteria. Some species even have various degrees of hybridization like grizzly bears and polar bears, forming growler bears. Yes, that's a real thing. Despite these limitations, the biological species concept will enable us to effectively talk about macroevolution without getting lost in the weedy details of exceptions. Like I said a moment ago, disruption of gene flow leads to speciation events between populations. Many populations have or develop something called reproductive barriers that cause them to become reproductively isolated from one another. These barriers can be divided into two primary groups: prezygotic barriers that take place before the sperm fertilizes the egg, and postzygotic barriers that occur after a hybrid of the two species is created. Let's take a look at these cool barriers in closer detail. Starting with the prezygotic barriers, our first one is habitat isolation. Habitat isolation occurs when you have two organisms that do not encounter one another because they have separate habitats they live in. The easiest example to understand would be a fully aquatic snake and a fully terrestrial snake. These two snakes, though they may be able to interbreed, are isolated from one another based on where they live. Thus, they would be separate species because they do not interbreed. Our next prezygotic barrier is temporal isolation. Temporal isolation occurs when organisms breed at different times of the day or month or even year. These different breeding seasons can separate organisms that could have otherwise have interbred. You can see this example with the frogs that are found here, where wood frogs, pickerel frogs, tree frogs, and bullfrogs all have separate mating times when they become reproductively active and sing. One innate benefit to this is that if you separate when you sing from all of the others, you don't get drowned out by the crowd when you're trying to find your mate. Our next prezygotic barrier is my favorite. It's behavioral isolation. Behavioral isolation revolves around being isolated based on particular courtships or different behaviors that are unique to a species. This can include things like sexual dimorphisms and has developed into beautiful behaviors that become species specific. There are thousands of examples of these behavioral isolations, but I'm going to show y'all my favorite two: albatross and lightning bugs. Let's take a look at albatross, who have this mating ritual that is incredibly specific.
Oops.
Wait for them to get back.
Now that behavior is pretty cool and kind of funny, right? Well, lightning bugs are another example of behavioral isolation. You probably didn't know this, but we actually have several different lightning bug species. Each species actually flashes its light in different patterns. You can kind of think of this like a specific language between the organisms of the same species. If you don't speak the language correctly, then you wouldn't be able to reproduce. Our next type of prezygotic barrier is something called mechanical isolation. Mechanical isolation is where the sexual reproductive organs of the organisms don't match. You can kind of think of this like a lock and key mechanism. Here's a cool example from the moth family that I studied in grad school. You can see all of these look very similar. So what would keep them from interbreeding? Well, one is actually the structure of the male penis. That's what you see over here. Notice that the structures are distinctly different. These morphological differences determine whether or not two organisms can interbreed. The last type of prezygotic barrier is gametic isolation. This isolation occurs at the gametic level. So sperm gets transferred. However, the egg cannot be fertilized because the sperm don't have matching receptors on the egg. Okay, so those are our prezygotic barriers. Now let's talk about our postzygotic barriers. Before we go into detail about the different types of postzygotic barriers, keep in mind that there's one easy way to separate them into the two groups. Prezygotic barriers are all before fertilization. However, postzygotic barriers are all after fertilization, so they all involve hybrids. The first postzygotic barrier that we're going to talk about is reduced hybrid viability. This occurs when organisms from separate species interbreed and produce a hybrid that does not survive. Our second postzygotic barrier is reduced hybrid fertility. A good example of this is a horse and a donkey. When these two organisms are bred together, they produce a mule with unique attributes of both organisms. However, the mule is sterile and incapable of reproducing. This is why horses and donkeys are considered separate species. The last postzygotic barrier is hybrid breakdown. In this scenario, a hybrid is created, but every generation after the first crossing results in offspring that are less and less viable in their environment. While we're talking about hybrid breakdown, I should also mention that the effect of mixing species can go in the opposite direction as well. Now, this is not a reproductive barrier, but rather would encourage two species to fuse. This is referred to as hybrid vigor or heterosis. Essentially, when two species interbreed, they produce an offspring that is better fit for the environment and is more robust. Frequently, this is an agricultural goal of taking two plants with different tolerances and crossing them in order to get a more hardy plant. Okay, now all of those are pretty cool. But where and how does speciation occur? Well, speciation can occur in two primary ways. Either when two species develop without being geographically isolated, called sympatric speciation, or when there is a geographical barrier that fragments a population, and this is called allopatric speciation. Allopatric speciation is the easiest to understand because there's a physical barrier to gene flow. This is incredibly common in fragmented populations with ephemeral environments like islands and streams. As species become genetically isolated from one another and different environmental selective pressures filter what is most adapted, populations can develop striking differences. Sympatric speciation is just a little more complicated. The easiest to understand is sexual selection. If mates become choosy and preferences develop, like preferences for a specific color phenotype, then a population can become fragmented based on these preferences, leading to a separation in gene flow. Another way that sympatric speciation can occur is where differences in habitat preference develop. If a population is represented by diverse preferences for where an organism lives, for example, high in a tree canopy or down on the forest floor, then they have the potential of getting isolated from one another based on these preferences and result in a reduction in gene flow between the fragmented populations. The last type of sympatric speciation that can occur is something called polyploidy. Polyploidy occurs when there is an error in the cellular replication cycle or the reduction cycle. and results in an organism having different numbers of chromosomes. When chromosome numbers do not match, organisms will not get the correct pairing of genetic information, and it can cause some organisms in the same location to become reproductively isolated from one another. You may think that polyploidies are actually kind of rare, but it's thought as much as 80% of the plants on this planet develop through polyploidy sympatric speciation. In all of these examples, we are really discussing whether a population is fragmented and gene flow is disrupted. Populations that experience these speciation events can develop in three primary ways. First, if a separation occurs between two populations and the environment they are isolated into is very different, then speciation events will be reinforced and the organisms will continue to become more dissimilar, eventually developing into different species. However, sometimes populations fragment in relatively similar habitats, and though they remain separated, they do not develop different characteristics between one another. They simply stay stabilized at the current differences. The last scenario occurs when hybrids can form between the two separated populations. Hybrids offer the opportunity for gene flow to be restored after fragmentation. If hybrids enable gene flow to resume, then the two populations will fuse back together, forming one population as a whole. Whoa, that is pretty cool, right? You may be wondering how rapid all of this can take place. Well, in short, thousands to millions of years. But the process can be gradual or occur rapidly and is referred to as punctuated. So what determines the pace? Well, strength of the isolation, the type of change that resulted in that isolation, and the environment the organisms live in. Major changes in morphology or rapidly changing environments tend to cause populations to change more rapidly. Well, y'all, that's it for this episode of Purple Bear Biology. Be sure to like and subscribe to continue the biology conversation with me as I release new exciting episodes. As always, thank y'all for watching, and see y'all next time.